morning. My name is Nicole Spear. I'm the project manager at the IMEDS team. Just thank you guys for taking an hour out of your day to join us on our March IMEDS community call. I wanted to take a moment at the beginning here to announce that we are going to be moving these calls from the third Thursday to the third Friday of the month, beginning in April. We work closely with some Mini-Sentinel investigators, and they've been unable to attend because they have a standing conflict. So we'd like to accommodate as many folks as we can get, and Friday seemed like a good call time option. So at the same time, 11 o'clock in the morning, and I hope it doesn't create a conflict for others, but we'll kind of monitor attendance, and if we need to make another change based on, if a lot of folks are all of a sudden unable to join, we'll think about doing so. But again, these are recorded, and we'll post them on the IMED site along with the presentation for you to view at any time. So just, we'll see how it goes. But look out for the invitation. It'll be the third Friday. It'll land on April 17th. And we're going to do that schedule going forward for a little while and see how it goes. Again, if you can just check your phones. I know last time we had some sound issues, background noise. We're going to try to start this out without muting everyone. If I do need to mute folks, we'll take a minute to do so, and then I'll unmute the presenters so that they can talk, but we won't be able to hear you if you have questions. Before I turn the presentation over to our presenters, I'd like to give Mark Cater just a few minutes to discuss any of the new happenings in the lab. Mark, do you want to go ahead?
Yes. So there's a couple of announcements that I would like to make about research lab. One is that I placed new vocabulary 5.0 CDM vocabulary on the IMEDS Reagan-Udall.org/vocabularies website, so it's available for that. It's actually not the new data, but it's a new format of the data. So the data is identical to the one that we had in November. And this just represents 5.0 ability for people to use CDM 5.0 within vocabulary. The second announcement is that some people are not aware that we have Mini-Sentinel format in the lab. We do have Medicare data currently, and we are working to Medicaid data, so that should be coming up shortly. So if you have any questions or needs regarding vocabularies or data sets that we have in Mini-Sentinel or OMOP CDM format, please contact me or Nicole, and we'll help you out. Thank you.
Great. Thanks, Mark. Okay, so with us today is Dr. Richard Boyce and Dr. Matthias Samwald from the University of Pittsburgh and the Medical University of Vienna, and they're going to discuss some of the research that they've been working on in the IMEDS lab. You can see the title on the screen in front of you or in the slide decks that were provided to you ahead of time. So we will be posting these again to the IMEDS website if you want to take a look at them at a later date. But without further ado, I'm going to go ahead and hand it over to Dr. Boyce and Dr. Samwald. Again, if I have to stop you, if we get some background noise, I'll let you know, and then I'll go ahead and do the muting and unmuting. So thank you.
Thank you so much, Nicole, and hello, everybody. This is Rich Boyce. I'm going to be co-presenting with Matthias, and so we'll pass the presentation back and forth. And I just want to first of all thank the folks who put together the IMEDS lab and maintain it, because we were able to conduct the pilot study within the lab, and so we're really excited. I think we're a little unique compared to some other presenters that have come so far because we're both academics who are really interested in coming into the medication safety field and tackling some of the problems that are important to the regulatory science community. And I think this study is sort of an early example of some things that may grow into some very interesting results down the road. Also just briefly.
Okay, I'm going to go ahead. Sorry, I apologize. I'm going to go ahead and mute the group because we are getting some keyboard noise, and then I'm going to unmute the presenter. So just give me one moment. I apologize.
The conference has been muted.
And Dr. Samwald and Dr. Boyce, are you able to speak?
The conference has been unmuted.
All right, guys, I apologize. I think I need to know their phone number before we got started. So we'll continue, and if we have more issues, I'll kind of monitor what their number is in the audio control. Sorry for the technical difficulties there. Go ahead. I'm sorry to interrupt.
That's okay. I just brought the number to you.
Okay.
Because now there's echo coming back on the line. So.
Okay. And Dr. Samwald, could you chat me over your number as well?
Unfortunately, I'm dialing in via Skype out, so I might not have a proper phone number.
The conference has been muted. The conference has been unmuted.
Yeah, now I don't see the phone number in the list. Okay, everybody take a minute. Look down at your phone. Please mute it if you can. We're going to have to continue on with it. I hope we don't get very much background noise. I apologize.
Okay, no problem at all.
Sorry about that.
The echo seems to be gone now, so we appreciate that. Okay, so basically the topic is on pharmacogenomics, and what I wanted to say is that both Matthias and myself, and we see also listed here Catherine Blajek, who's one of Matthias's students and mentees. We're all very interested in developing pharmacogenomics clinical decision support systems and information resources that support the clinical workflows around pharmacogenomics. And so to some extent, the research we're talking about complements other initiatives that we are working on. And when it comes time for the discussion, we may point you to some of those other resources if you're interested. So in terms of significance, adverse drug reactions, I don't think I have to tell this audience that they're a major issue. Everyone on this call, I'm sure, is deeply interested in this topic. But maybe you haven't heard quite so much about how pharmacogenomics could help reduce the occurrence of adverse drug events. Principally, it's useful for personalizing a therapy. So, for example, warfarin, which is a drug commonly prescribed for anticoagulant purposes, has a very narrow therapeutic range and is often difficult to titrate an individual to the proper dose. And over the last 10 years, it's been identified that there are principally two proteins or enzymes that are responsible for a significant proportion in the variability that occurs when patients are trying to reach a clinically acceptable dose. And there are pharmacogenomics testing available. There is pharmacogenomics testing available that, when conducted, can help to narrow down the range of a starting dose and hopefully reduce the amount of time it takes to bring an individual to the appropriate dose. Similarly, another drug like clopidogrel, which is often used as an antiplatelet for cardiovascular therapy treatments, also is susceptible to a pharmacogenomics pathway and leads to variability in the actual effectiveness of the drug. So there again, knowing about the person's individual phenotype, as they would call it, and adjusting the dose to the appropriate or recommended amount can help improve the effectiveness of the drug and reduce adverse events. So the other thing that I would say about this, and I will actually paste this into the chat, is there's about 131 or so drugs right now that have some kind of pharmacogenomics recommendation or knowledge around it. And I'm trying to actually, let's see here, the chat's a little bit harder to do than some other places. So if you're able to receive that chat, that's a table that's up on the FDA website, and it lists the actual drugs and biomarkers for which somewhere in the FDA-approved drug product labeling there's a discussion around clinical pharmacogenomics. So there's a lot of drugs for which this sort of thing could be relevant, but there's a lot of also concerns around how to fit this into the workflow, and is the return on investment for actually conducting these tests going to be there. And so going on to the next slide, really we wanted to consider two different scenarios. One would be a preemptive pharmacogenomic testing scenario, and that's where everyone in a health system's population would be tested for some panel of pharmacogenes, as we will call them. And then that information would reside in their medical records, and all future prescribing could take the knowledge of the patient's pharmacogenomic phenotype into account. A second scenario would be one that we're calling the reactive pharmacogenomics phenotyping, and that would be where a panel is only actually initiated when the first time that someone receives a drug for which is a pharmacogenomics recommendation. Now, up until this point, there have been very few studies that have actually collected data that would be useful for determining if there would be a return on investment. What's needed is the incidence of exposure to the drugs for which is a pharmacogene, and also the knowledge about the rate at which adverse events associated with that drugs occur, and some estimate of the likelihood that knowledge of the pharmacogene could reduce the incidence of the adverse event occurring. So we were going to tackle the first problem, and that is to focus on the incidence of exposure. And so basically our goal was to use large claims databases, which have never been used to ask this question, and look at how many people within a certain time period will be incident users for at least one drug for which is a pharmacogenomics recommendation. But we also extended the analysis to look at how often will they be exposed to two or three or four drugs for which is a pharmacogenomics recommendation. And also this analysis could kind of prioritize which drugs are the most likely for them to be exposed to. And the nice thing about the large claims databases that are available in the IMEDS lab is you have a range of populations. So you have very young people in sort of the Truven CCAE data set, all the way to folks who are older than 65 in the Medicare supplemental data set. So we conducted the analysis, as Matthias will explain in more detail, and then we stratified the results. And so the idea then is the results should be useful to other health systems who will then, conduct their own analyses on cost benefit. So at this point, I'm going to turn the controls over to Matthias, who will describe the methods and the results.
Hello, this is Matthias Samwald from the Medical University of Vienna. I quickly go through the methods that we used and also present some of the key results that we got. So at the outset of our study, we looked at the available clinical pharmacogenomic guidelines that were already available. And internationally, there are two major sources of such guidelines. In the United States, there's an organization called the Clinical Pharmacogenomics Implementation Consortium, or CPIC in short. And there's also another group in Europe, in the Netherlands to be precise, called the Dutch Pharmacogenetics Working Group, or DPWG. Just refer to them as the Dutch group from now. And those are the two major groups that have worked on creating detailed guidelines for pharmacogenomics, how drugs should be dosed or which drugs should be selected based on genetic markers. And so besides the geographic difference, these two sources also differ in terms of coverage. So the Dutch group at the time of when we started our analysis, they had already created guidelines on 73 substance gene interaction pairs. So in total we looked at 73 substance gene interaction pairs, and of those pairs, 25 interaction pairs were described by CPIC, and 62 interaction pairs were described by the Dutch group, with a major overlap, of course. But in general, the Dutch group is more inclusive and has more guidelines available, while CPIC, the group from the United States, is requires higher standards, at least that's what they claim, for inclusion, and they have also looked at a smaller number of drug-gene interactions so far. Basically, if we look at the key statements made in these guidelines, they are mostly about drug dosage, adaptations based on the capability of individual patients to metabolize certain medications. But in some cases, there are certain strong recommendations that actually tell practitioners to use another medication altogether. So here we see listed two excerpts, one from CPIC about clopidogrel, where based on CYP2C19 poor metabolizer status, another therapy is recommended instead of clopidogrel. And we also see an example from the Dutch group where the dosage of escitalopram is adapted based on the ultra-rapid metabolizer status of a patient. So in terms of drug prescription data, we used the IMEDS datasets, of course, and we used 3 different datasets. First, the CCAE database of privately insured patients covering over 100 million patients. Then the Medicaid dataset covering over 50 million Medicaid enrollees. And finally, the Medicare dataset covering 8 million U.S. retirees with Medicare insurance. And these datasets in general cover, as most of you probably know, a time span from 2003 to 2012, 2013, roughly that. And we used the Common Data Model and the Standard Vocabulary version 4, and used the SQL workbench to query the database. So nothing magical happened here. And so the basic design was actually quite simple. So it was a cross-sectional study of the drug utilization across each dataset. And what we tried to look at was the number of incident prescriptions in a defined time window. So we defined a time window of 4 years, starting in 2009 and ending in the year 2012. And we looked at the incident prescriptions of medications that are listed in one of the 2 guideline sources that I've described earlier. So we're only looking at medications that were either described in a CPIC guideline or in a guideline from this Dutch working group. And we only looked at incident prescriptions. So if the data in the databases indicated that a certain patient was already prescribed a certain medication prior to this 4-year time window, this was excluded. So we based on the reasoning that if a patient is already being prescribed a medication for several years and doesn't have any issues with it, then his or her prescription would probably not be updated by data on pharmacogenes anyways. And we also excluded topical preparations of pharmacogenomic drugs, because usually, actually in all of the cases, the guidelines only refer to oral or other systemic preparations, but not to topical preparations. So, as I said, we first divided the data into these 3 data sets, CCAE, Medicaid, and Medicare. And then we also, for each data set, we created age cohorts, so from 0 to 13 years, from 14 to 39 years, and from 40 to 64 years for the CCA and AE and the Medicaid data sets. And for Medicare, we only had the patients with an age over or equal to 65 years of age. Okay, so this is how we divided up the data sets. And as Rich already said, we also compared two different testing settings in our simulations. One being the preemptive testing setting, where theoretically all of the patients would be tested at the start of this 4-year time period, and then we would check how many patients would be prescribed drugs that could be optimized by a test that would cover all of these medications in the clinical guidelines, and then also so-called reactive preemptive testing scenario, where a genetic test would only be conducted at the time of the first incident use of one of these pharmacogenomic drugs. So this would be a slightly more targeted testing approach than just testing everybody at the start of this 4-year period. And of course, we are well aware that these are highly theoretical scenarios, but what we wanted to do was to check what the maximum number of different medications would be that could be optimized by a test that would, so to say, be able to get generate data that could be used for all these pharmacogenomic medications that are already captured in pharmacogenomic guidelines. So in total, we considered 61 drug substances. So those is the union of the drugs mentioned by CPIC and the guidelines from the Dutch group. And these 61 drug substances participated in 73 substance-gene interaction pairs. Looking at the guidelines, we also saw that not all genes and not all interactions were created equally, and we divided up this list into a core list and, well, another list that is of drugs that do not belong to the core list. And we defined the core list as those drugs that could be optimized by having data on those genes that are typically covered by established pharmacogenomic preemptive genetic tests. So this would be genes that are typical cytochrome genes like CYP2C19 or CYP2D6, very common genes that would be tested in pharmacogenomic panel assays. But then we also saw that some other genes were listed that would not be on this list, and yeah, we'll look into this in more detail afterwards. So roughly over 89 million patient records were included in this analysis. 55.3% of these patient records were associated with female patients, and on this slide you see statistics about the prescription of multiple pharmacogenomic drugs within this selected 4-year period. So of course we can only show a fraction of the data and of the statistics we generated as part of the study, but these statistics might be the most relevant because it doesn't only show you how many patients received some kind of drug that is listed in some pharmacogenomic guideline, but also how many patients received multiple different drugs within this 4-year period. And this is, of course, especially of interest if we are trying to argue that a preemptive genetic testing approach would be superior to an ad hoc reactive approach where you just test one single gene for one single drug when the prescription occurs. We want to show that, so to say, with one test that includes all of these pharmacogenes that are already known and are already described in clinical guidelines, we are then able to optimize treatment of a multitude of different medications in the years after the genetic test was done. And you can see this here in this table. As I said, we see the fraction of patients that received 2 or more PGX drugs as incident prescriptions within this 4-year period. And we see this split up by age and by data set, by insurance provider. And not surprisingly, we see that older patients are more likely to receive a multitude of different drugs that could be optimized by pharmacogenomic data. So for example, for the Medicaid cohort with the ages 40 to 64, roughly 32% of the patients had incident prescriptions of 2 or more of these drugs where a test could be used to optimize the treatment. And also for Medicare, we see that roughly 27% of the Medicare patients received 2 or more drugs. So this, so to say, would be the fraction of patients where, that would be tested in a purely preemptive scenario. You take all of the patients you have in your system, test them with a preemptive pharmacogenomic test at the start of the 4-year period, and then, roughly 32% of this cohort would receive 2 or more drugs that could then actually be optimized with the data you generated in this test. In the next slide, we also see the data for this other scenario that I described, the so-called reactive preemptive, where preemptive testing would not just be handed out to all of the patients, but in a more targeted manner, where the test would only be done at the moment where the first pharmacogenomic drug would be prescribed. And of course, we see that the fractions of patients that would then receive 2 or more pharmacogenomic drugs is rising significantly, because we are losing all of the patients that haven't been prescribed any pharmacogenomic medication at all. And we see that, for example, for the Medicaid, for the older Medicaid cohort, almost 60% of the patients would receive 2 or more PGX drugs, and for Medicare, almost 55% of the patients would then receive 2 or more such pharmacogenomic drugs. And all of this data were for this core list. And as I said, we excluded some of the genes because we found them not to be usually among the genes tested by pharmacogenomic assays. And these genes included, for example, F5, factor 5. This gene is listed by the Dutch group for patients that for female patients that receive oral contraceptives, and we have a guideline that cautions against using oral contraceptives for certain variants of this gene because of an increased risk of thrombophilia. And then there are some other genes like HLA-B, which is relevant only for very rare adverse drug events, but for very severe rare adverse drug events. And including these other genes in the analysis as well, of course, also increases the fractions of patients that would be identified for optimized treatment by a preemptive pharmacogenomic test. Of especial interest in this regard is, of course, F5 and the oral contraceptives, because a large fraction of the female patients in the data sets receive oral contraceptives at some point in time. And this basically shows that as the number of the gene-drug interactions in the guideline grows, the number or the fraction of patients that could potentially profit from pharmacogenomic testing can also grow quite tremendously. If you include oral contraceptives, all of a sudden a large fraction of the patient population could profit from such a test. And this is something that our studies showed, that choosing the drugs and choosing the guidelines that you include can tremendously impact on the fraction of patients that could profit from a genetic test. We also looked at the medications that could be optimized in this way, and we saw that especially for younger patients, analgesic medications and also neurological medications made up a huge share of the medications that could be optimized by pharmacogenomics. This slightly changes for older patients, and especially for patients over the age of 65, we see a more balanced distribution between therapeutic classes, so that cardiology medications or endocrinology medications make up a larger share of the medications. And of course, all of this is just a very preliminary glimpse into the complexity of the data and the potential that pharmacogenomic testing provides. And with this, I hand over to my colleague Richard Boyce, who will talk about future work that we plan and where we also want to invite you as potential participants. Thank you.
Thank you very much, Matthias. So I think the importance of the study is that, to our knowledge, this is the first analysis to use large claims data sets to examine the potential for preemptive pharmacogenomics testing by examining the incident exposure in different patient populations. Prior to this, for example, the largest study was probably done by Predict at Vanderbilt in their community population, and they only had about 10,000 patients and a much smaller set of drugs and pharmacogenes that they were testing for. So this contributes knowledge about the variability across populations and age groups. But with that being said, we know that other health systems will have even different populations, and just incidence data alone is not sufficient to conduct a rigorous economic analysis. And so we wanted to find a way to extend this analysis, and one of the things that we've done is we've created on in the Odyssey Collaborative a research protocol, and I'm going to paste that into the chat here for everybody. So the idea of this research protocol is to kind of conduct the same incident analysis, but expand the participation to other health populations. And in addition to that, while the protocol right now is in development, so if there are other aspects that we would want to look at, for example, Susan Gruber, when she saw the early results on this, she pointed out that there seems to be possibly a higher likelihood of being on a second on certain there seems to be a higher likelihood of a patient receiving a drug for which is a pharmacogenomics recommendation once they've already received another one, and that there may be certain drugs for which that increases the risk even more. So there may be basically covariation associated with the actual drug that the person is being exposed to. And we did no analysis at this point of covariation or the risk factors for exposure to these drugs, and so perhaps that could be a part of the protocol as we move it forward. And so there it is again, just mentioning that. And with that, we have time for questions. I just want to acknowledge the Reagan-Udall Foundation for allowing us to conduct this study. Matthias is funded by the Austrian Science Fund. I have been funded by the National Institute of Aging, National Library of Medicine, and we have other colleagues associated with this project. Catherine was mentioned on the title slide. Here at the University of Pittsburgh, Phil Empey in the School of Pharmacy has contributed. We've had people who have contributed also, including Patrick Ryan, Sayed Musa Ahmed, Ashton Hoffer. So we thank all those folks and the people who help provide the funding for this. So with that, we'll open it up for discussion. Thank you very much.
Thank you. Thank you. We are getting a little bit of feedback, but it might be coming from your end. Not sure. Okay, great. I think Alec actually muted himself. Okay. Thank you guys so much, and also thank you for at the beginning giving props to the IMEDS lab. That's an important thing, and there's a lot of interesting work going on in there, and we just appreciate you taking the time to do that. So if you want to just unmute yourself now and announce yourself, feel free to ask away. Any questions or discussions?
Yeah, hi, this is Susan Gruber. I guess I have a follow-up question about how you chose those age categories. Is there some biological significance to that choice?
Matthias, if you don't mind, I'll go first and then chime in. The first breakdown that we wanted to make was between the older population and the younger population, and that's motivated by the fact that my research interests are in medication safety for older adults. So it was more driven by my actual research interests. But then other people on our team, including Matthias and Catherine, expressed that some of these other breakdowns would be really interesting. And as we saw, the breakdown for the adolescent age period is actually really interesting, because that's when you see a spike in the ethinyl estradiol, which has that unique recommendation, and that's the time period when it actually spikes quite a bit. So the age breakdowns we chose were more motivated by kind of our research interests.
Thank you.
This is Alec Walker.
Great, Alec.
It seems that when you look to the future and you're offering for users to do cost-benefit analyses, you're asking them to step into a real swamp, because the frequency of events is variably quantified and is different in different populations. I'm wondering if you couldn't do a simpler step by identifying drugs for which testing is recommended. For example, with oral contraceptives and older women, factor V Leiden testing probably is indicated. And just do it on a yes-no basis. These are people for whom there is an indicated testing that's generally accepted in practice. And then view your frequencies around that, so that you don't kind of ask people to do a lot more research in order to utilize your data.
Alec, I think that's a very good suggestion, and I appreciate that. In another project, we've actually been annotating the drug product labeling, referring back to that FDA table that had the 130 or so drugs with pharmacogenes. And one of our annotation fields is testing recommended. So I think that would be one source that we could pull from. One of the things that we are aware of, because like here at Pitt, for example, we're doing a pharmacogenomics pilot, is that the local health systems, oftentimes they have their priorities and they want to develop their own localized guidelines. There's not necessarily a lot of trust for outside guidelines such as CPEC within a local health system, let's say. So that kind of gets back to the swamp you were talking about. Definitely we think that for the majority of the drugs with pharmacogenes, the further analysis that needs to be done is going to be driven by their own local sort of care practices and priorities. But I really like your idea, and I wanted to ask you, do you have a suggestion for other sources that we could look at for test recommendation? I know the PharmGKB would be a good source, but outside of the PharmGKB and drug product labeling, do you have any ideas?
Well, no, I don't have a source to direct you to. Let me give that some thought. I mean, I think that, not even experienced clinicians. But so the answer is no, I don't. There are a couple like factor V Leiden older women that I had personal experience with that I know about. And I think you're just going to have to keep asking around, but nothing more than that. Sorry.
Thank you, though. Great suggestion. I appreciate that.
Great. Thanks, Alex. Got plenty of time, so if anybody has anything else they'd like to bring up, feel free to do so.
Yeah, this is Alec again. The other place to be thinking about here is, in a sense, you did one demographic cut, which was by age. Something that would be straightforward to do would be to look for other simply easy to identify markers of high prevalence of use of these drugs, and certainly particular diagnoses. A lot of the drugs for which testing is required are oncology drugs, so that people had kind of a series of rule of thumb that they say, okay, this person is in a population I need to look at more carefully.
So I think I understand because even with the reactive pharmacogenomics scenario, there's this it's hypothetical, and in actual practice, you would have to order the test at the time of prescribing, which there would be a delay. And how practical is that in a lot of situations? Whereas what you're suggesting is, disease categories, for example, where that there's probably, or where we could determine if there's a high likelihood of being prescribed one of these drugs with pharmacogenes, and using that as the trigger for the order.
Yeah, and you had some evidence that would be fruitful if you looked at the older Medicaid patients, that is the 45 to 64s. That's a very different population from the younger ones. That's heavily spiked with people who are in there because they're poor because of chronic disease, and you're seeing, not surprisingly, a lot more drug use in those groups. And similarly, if you looked at Medicare patients younger than 65, that's also people with renal failure and things like that. I'm sure you'd find very high prevalences of these drug use. But just looking for just kind of simple markers that you could tell clinicians, boy, people in this category deserve preemptive testing.
Well, I think we should invite you to participate on the protocol development for the network study that we're talking about, Alex, since you've already made some great suggestions.
Thanks for the offer. I'm glad to offer advice, but sure, we'll talk more.
Great. Okay, anyone else? Okay.
So could we just ask opinions in the community around the pharmacogenomics or personalized medicine studies in general, and how they might be of interest? What other kinds of studies might be of interest to this community? Does anybody want to suggest or ask some questions around that?
Are there currently reimbursed studies that appear in these data files you've been looking at as pharmacogenomic assays?
You mean, like, are there codes for reimbursement for the tests?
Yep.
I just heard that Medicare approved certain billing codes, but I don't know what they are, so I need to look into that. Not in the data that's currently in the IMEDS lab, because it was a recent development.
Hi, this is Patty St. Clair. I just wanted to reiterate Alex's comment on the younger Medicare patients. You may find that they're also Medicaid, and so you may pick up, if you look at their drug fills, you may pick up more among the Medicaid patients who are younger. I missed some of the beginning, so I'm not sure if you did that, but if you scanned both Medicare and Medicaid, the dual eligibles that are young are potentially disabled, and I think a fair number of them will show up with some of these psychiatry neurology drugs, as you're seeing in the Medicaid population anyway. So not so much a question, but just I guess the question is, did you include the how did you handle the younger Medicaid and also Medicare eligible patients?
Thanks, Patty. So you mean the dual eligibles? I guess what I think I hear you asking is, did we try to link between those two insurance plans? Is that
I see. Yeah, I see. Yeah, did you link people who might have changed over from Medicaid to Medicare at some point?
That's not possible with the Truven data sets. Maybe someone else could comment.
Okay. Thank you.
I would say this too. The Medicare data set is the supplemental for employers. So this is employer-provided supplemental insurance, which I really think would be a very different population than the general Medicare population. That currently is not present in a common data model version, though within the Odyssey community they're working on that. There's a simulated data set for Medicare with a certain percentage sample. I don't know what it is off the top of my head, but they've been working on doing the ETL to bring it into the common data model. So sometime hopefully over the next year, there will be at least a simulated Medicare population that's more generalizable than the supplemental insured.
I see. Okay, great. Thank you.
Okay. Any other questions or comments from the group? And Dr. Boyce or Dr. Samwald, is there anything else you'd like to ask the group from your end?
No, I want to thank everybody for their time and for attending our talk. Just as a follow-up, I'll paste into the chat the two projects that we're working on for folks' interest, and they'll take you to links where you can learn more about our pharmacogenomics-related research. But again, thank you so much for your time and attention.
No, thank you guys. Thanks both of you. In fact, what we'll do is maybe we'll even try to link that through our website too. So if people want to take a look at the presentation, we'll try to have that information available for you up there as well. And thank you both for taking time out of your day to present to us. We really appreciate it. Sorry for the technical hiccups in the beginning. Every time this happens, we end up learning how to fix it by the end of the call. So we'll just keep getting better. And again, the next call will be on Friday, April 17th. We'll send out an invitation shortly with the topic and the agenda for that call. And just thank you guys again. We really appreciate you attending these, and have a great day.
Thank you.
Yes, thank you. Great talk. Thank you.
Thank you, guys. Bye-bye.
Thank you.